Hi, Lilith again. This is a continuation of the video before this. So in the previous video, we finished talking about beta oxidation and how in beta oxidation, fatty acids are broken down into acetyl-CoA molecules. Right, and because acetyl-CoA molecules are 2 carbon molecules, you can see how an even numbered carbon chain in the fatty acid will be evenly broken down into different molecules of acetyl-CoA. However, we will run into some problems if we have an odd numbered fatty acid. But also, we will run into some problems if the fatty acid is not fully saturated. So let's talk about those two problems and how the cell takes care of them, right? The problem of having an unsaturation in the chain, and also the problem of having odd number of carbons in the fatty acid chain. So if the fatty acid or the acyl-CoA has an unsaturation and it is a cis unsaturation, then the cell will use the enzyme propionyl-CoA isomerase to turn this cis unsaturation into a trans unsaturation. And this step is absolutely needed because if you look closely, this molecule is now an intermediate in the beta oxidation. Let me zoom out to show you exactly where. Right here, you see how we have that trans unsaturation. So when the enzyme propionyl-CoA isomerase turns the cis unsaturation into trans, then this molecule can now easily go into beta oxidation and get broken down to acetyl-CoA molecules. Now, how does the cell deal with having odd numbers of carbon in the fatty acids? Well, imagine we have a fatty acid that has 9 carbons. What happens is we start to cut away on the fatty acid this direction. Actually, this isn't a free fatty acid, right? It should be with a CoA. So let me fix that. There we go. So what happens is, first, these two are cleaved from here, then these two are cleaved from here, then these two are cleaved from here, and at the end we are left with 3 carbons. And that 3 carbon leftover is what propionyl-CoA is. It is the last 3 carbon acyl-CoA that cannot be broken down by beta oxidation anymore. So, to not waste these carbons and to still make it useful for the cell, propionyl-CoA carboxylase uses biotin and bicarbonate and ATP to carboxylate this middle carbon right here. And when it does this, you get methylmalonyl-CoA. This molecule then undergoes a reaction catalyzed by methylmalonyl-CoA epimerase, which epimerizes the molecule into methylmalonyl-CoA. Well, yeah, L-methylmalonyl-CoA from D. Then, methylmalonyl-CoA mutase uses coenzyme B12 to form succinyl-CoA. And as you already may know, succinyl-CoA is an intermediate of the Krebs cycle, so it can easily enter the Krebs cycle and continue being processed there. So you see how even though a cis unsaturation is a problem, right? Air quotes, problem. And having propionyl-CoA is a problem also. The cell has ways to still make everything happen, basically. To take a closer look at coenzyme B12, it's important because it has this cobalt in the middle of it. And what cobalt does is it is involved in radical chemistry. And a radical is 1 electron. And since a sigma bond is made out of 2 electrons, when a sigma bond is cleaved, you could be left with a radical. So what coenzyme B does, coenzyme B12, it uses radical chemistry to break and form sigma bonds and be able to move this group onto here. Next, we can discuss ketone body formation. So ketone bodies are formed when we have a surplus of acetyl-CoA molecules. And this is important because if we have broken down a lot of glucose and we've generated a lot of acetyl-CoA, or we have broken down a lot of fatty acids and we've generated a lot of acetyl-CoA, and that has taken place in the liver, then the liver must have a way of being able to send those acetyl-CoA groups to other tissues in the body. And it does it by packaging the acetyl-CoA groups into these ketone bodies. And the 2 ketone bodies that we talked about are hydroxybutyrate, or beta-hydroxybutyrate, and acetoacetate. So let's look at how each of them are formed. We take 2 acetyl-CoAs, and through a Claisen condensation reaction, the enzyme thiolase makes acetoacetyl-CoA. Then the enzyme HMG-CoA synthase produces HMG-CoA, or beta-hydroxy beta-methyl-gluteryl-CoA. And then HMG-CoA lyase cuts away 1 acetyl-CoA group, and we are left with acetoacetate, which is one of the ketone bodies. Later, an NADH can be used to reduce acetoacetate and form beta-hydroxybutyrate, which is catalyzed by this hydroxybutyrate dehydrogenase enzyme. If you've read the name game, then it should be fairly easy for you to understand why this molecule is named the way it is. Same for this, same for this, same for everything really. So make sure you do the readings because it will only benefit you. Now that we've formed the ketone bodies, acetoacetate and beta-hydroxybutyrate, now these ketone bodies can travel throughout the body to different types of tissues. What happens when they get to the tissue? Well, now that tissue, the cells in that tissue, must be able to use these ketone bodies. So, one of the ketone bodies, beta-hydroxybutyrate, is taken up by the cell. It is oxidized by NAD+. Oxidation happens over here, and we form a carbonyl acetoacetate. Then, through this transferase enzyme, acetyl-CoA lends its CoA to acetoacetate and forms acetoacetyl-CoA. And thiolase uses a coenzyme A, to break this apart and form 2 acetyl-CoAs. Now, this tissue that had received the ketone bodies has 2 acetyl-CoAs per ketone body. And now they can shunt this away into the Krebs cycle and be oxidized even further, produce NADH, FADH2s, ATPs, and further on. This is the end of this short video. If you have any questions, please don't hesitate to ask. These notes are located in my IATA folder at the very end of our Canvas page. So that's where you can find them. And good luck studying. You all got this, and everything will be okay.